Hello everybody, in this video we are going to talk about viral replication. Yes, in this video we are going to talk about viral replication. And as you already know till now, viruses are not completely living. They cannot be told to be completely living. Why? Because they have no organelles. Neither the membrane bound organelles nor the non-membrane bound organelles. They have nothing. That's why viruses cannot reproduce through the general means like asexually or sexually. They cannot do that. Why? Because they cannot copy their genetic material only. For that, we need specific proteins to copy the genetic material. If you know the process of transcription, then you know that copying the genetic material requires special type of proteins. Also, viruses cannot make proteins of themselves. For the capsomers, the units of the capsid layer present outside the virus is made up of proteins, right? And they, to make another virus, they need to have those proteins. They need to build up the capsomers and then join together and make the capsid layer, but they cannot build up the capsomers, so they cannot replicate by themselves. They have to be parasitic on any other cell or any other organism, so that they can use the machinery of the organism to replicate themselves. So that's why viruses are obligate parasites, as I told in the previous video. Okay, so in the previous video we talked a little bit about the cryptograms and the basics of viruses. So if you don't know about that, you may refer to the previous video. You will find the link over here somewhere. So let us dive into the process of viral replication or the reproduction in viruses. That can be of two types, majorly of two types. Alright, and what are those two types? First of all, it is the Lytic cycle. Where's my, yes, smaller than that. Okay, the Lytic cycle or Lytic cycle, whatever you want to pronounce. Lytic cycle. And the second one is the Lysogenic cycle. Lysogenic cycle. Okay, so that was the two types of major viruses, reproduction procedures of viruses. But why am I telling the word major in every time I am telling about this? Because as you may have guessed that not only there are two types of viral replication processes, but there are a third category of viral replication process. As you know, biology is full of exceptions. So that third type of viral replication process is neither completely lytic nor completely lysogenic. It is both. First of all, it becomes lysogenic, then it becomes the Lytic cycle. We will discuss about that type in some other video, and those viruses which show that type of viral replication are known as retroviruses. Example, HIV, as you may have heard the name of. It is the human immunodeficiency virus. Yes. So let us dive into Lytic and Lysogenic cycle. So first of all, any virus to gain control over the organism or the host, they need to enter the host, right? So there can be two ways in which they can enter the host. Let's say this is the cell. Okay, and here is our small tiny virus which wants to gain entry in the host, and the virus will have a genetic material and the cell will also have a genetic material. Okay, let me see how well I can make a genetic material for the cell. Okay, let's say it is a circular RNA, and here is our virus with a single stranded RNA. As you can see, anything can be there. Now the virus wants to get inside that cell. Alright, so there can be two types, two ways the virus can choose between those two ways on how it wants to enter the cell. One way can be receptor mediated endocytosis. Sounds like a mouthful, but let me explain it. Here it is some receptors in the cell membrane of the cell. Pretty good till now. What are the receptors used for? There may be some nerve impulses in a multicellular organism. There can be the nerve impulses coming, and then the nerve impulses are felt by the cell through these receptors. In unicellular organisms also these receptors are used. They can be used as sense organs, as we have our eyes. The unicellular organs can have their receptors and they can sense about the environment. So they normally have receptors in their cell membrane. Not only few, they are in a few places also. And when the virus doesn't have a lipid envelope, it has to enter the cell through this receptor. So it is called receptor mediated. Now, why it is endocytosis? Anything which is gaining entry into the cell is in the form of a body. Alright, it is enclosed by the cell membrane. So, whenever it is going inside the cell, it is enclosed by the cell membrane, it is engulfed by the cell, or it is making the cell to engulf itself, the virus. Okay, so that's why it is called endocytosis. Endo means inside, cytosis means the cell. So it is going inside the cell, that's why endocytosis. Okay, lot of it. So this virus will go here, okay, and it will sit over here in this receptor, and it will give some signals so that the cell will feel that it is some useful thing. So it will try to engulf it, or will try to take it in, and then the virus will actually go inside the cell in the form of a body. So there will be the virus, okay, and there will be some part of the cytoplasm, because the cell has taken it in the form of a body, endocytosed it, and the RNA material will also be there. So pretty good till now. There are two ways I told. This was the first one. What is the second one? Second one is only for the viruses which have an envelope outside them. So let's say the virus has an lipid envelope outside them. Okay, this is the envelope, and in this way, in this type of viruses, they have another choice. They can enter through this receptor mediated response also, but they can directly fuse with the cytoplasm also. So what will happen? The envelope will fuse, and it will form a body inside the cell. So like that it will fuse, and the envelope contains a capsid as always. So there will be the capsid here, and the genetic material also. So like that. So this is the cell directly fusing with the membrane and then entering the cell. This is the virus directly fusing with the cell membrane and then entering the cell, and this is the virus entering through receptor mediated responses. So why can only the viruses with the envelope be able to do this type of entry process? Why cannot the viruses with a capsid layer? And the answer is very simple. What is our cell membrane made up of? It is a phosphobilipid layer. So lipid, and here the envelope is also made up of lipid. So the thing is that to fuse two things, they must be made of the similar kind of, okay, they must be similar kind of. So this is also lipid layer, this is also lipid layer, so they can fuse. But the capsid is a protein layer made up of capsomers, and this lipid layer, that is the envelope, is made up of peptidomers, for your information, and this capsid cannot fuse with this membrane because it is made up of different things, right? So this has to maintain this receptor mediated process. So up till okay now? Okay, now we will move on to the next step, where the viruses has entered the cell. Let me quickly make the diagrams. Okay, the cell will be black, and there will be our virus. Where is the black virus? Okay, there is our virus. Do not care about the shape here. I'm drawing any irregular kind of shape. Okay, and the genetic material of the virus, here it is. And I forgot to draw in the previous step that, yes, I drew it. The cell will also have a genetic material, and here is the cell's genetic material. I'm drawing it in green, let's say. Okay, here is the cell's genetic material. Now as it has entered, what happens is that the envelope outside it, or the capsid outside it, anything outside the genetic material of the virus, just dissolves inside the cytoplasm. So all these things, let me show it in brown, dissolves like that. All these outside things dissolves. Okay, they dissolve out. Dissolve. Okay, and we have our naked DNA or RNA strand of the virus inside our cell. This is the second step. What happens in the third step? Let us see. In the third step, we have our cell as always. Where is our, yes, we have our cell as always. Let me make it black, and then the genetic material of the cell, I am showing it in green. I am showing the genetic material of the cell in green. But okay, I will tell it later. And there is the genetic material of the virus in red. Now the genetic material of the virus will attack the genetic material of the cell and destroy it completely. So you might think that what is the need for destroying the genetic material of the virus, genetic material of the cell? What is the need of doing? The need is that due to the presence of the genetic material of the cell, the machinery of the cell will not listen to this foreign genetic material of the virus. Why will it listen? It will just code and translate the genetic material or the information of the genetic material of the cell, not the information of the genetic material which came from the virus. So it needs to get some importance right in the cell, so it will destroy it completely. It will tear it up in pieces, and then this genetic material will be useless. Then the cell will only have the viral genetic material, and then it has to translate those genetic material only. So that's why, to make that happen, this genetic material of the virus will destroy the genetic material of the cell. That was the third step. Alright, now let us move on to the fourth step. Fourth step, what happens? Here is our cell. This time it is already in black, and this genetic material of the virus has destroyed the genetic material of the cell. So it has broken down into pieces like that. Let me see, small pieces. Okay, it has been broken down. Genetic material of the virus is still intact, and it will replicate itself. Now the cell's machinery will listen to this genetic material, and it will help it to transcription process. That will make copies of itself. Okay, more copies. Now this genetic material will be translated also. So the translation means from the cell's machinery will read the information present in the genetic material and then code proteins and make proteins from it. So it will get the proteins of the capsomers. So it will get make the capsomers and make the capsid layer too, and it will make the envelope also if it has one, if the virus has one. So there will be an envelope. Where is, how can I show it? Yes, there will be an envelope. Okay, so the virus will make all the parts of itself. Okay, till now all right. There will be envelope, there will be capsids, there will be genetic materials. Now what happens in the next step? Yes, as you have guessed, you are right, that in the next step all these parts will be assembled together, and when these parts will be assembled together, there will be more than, well, it depends upon the size of the cell. There will be so many viruses which are generated from one virus. So where is my pencil tool? Over there. Yes, and let me take this color. Yeah, okay. Now let's see. This has made its capsid layer. It has made its don't pay attention to the background noises. It has made its what we say, genetic material. It has made its single stranded RNA, as you can see, and it has also made its envelope. As I have shown the envelope in the previous step, I should show it over here also. And not only one, there are many such viruses which have been produced like that. So let me, for simplicity, copy and paste the thing. Okay, ctrl-c, what I did. Alright, and v, bring it over here. V, bring it over here. So like that all viruses will be formed. What I am doing, this is very bad. Yes, bring it over there. Okay, like that many viruses will be formed, and it will keep on happening, this process, until the space in the cell becomes so short that it cannot hold more viruses. Alright, so what will happen? Eventually the cell will burst out and the viruses will leak out, and ultimately the viruses will go out of the cell, and then the viruses will affect more such cells, and then all the same things will happen. So that's why it is a cycle. Alright, so this is a cycle. What will happen now? The cell membrane will be destroyed because more and more viruses are keeping to be produced over here. It cannot hold so many viruses, so it will just burst out like that. Let me show it the best way I can. Okay, it has bursted out, and the viruses which will be produced are leaked out like that. The viruses are leaked out, and there will be neighboring cells if it is a multicellular organism, or if it is a unicellular organism, they may be colonial, so there will be some neighboring cells which the virus will again attack, and then again do the same processes, and then it will replicate itself more and more. So that was the process I told is the lytic process. That is only one of the lytic process. So what do you think about will be the lysogenic process? The lysogenic process is not much different, but a little more steps added to the Lytic cycle. Sorry, it became the red color, but I hope you get the general idea. So where does the difference between the Lytic and lysogenic cycle occur? See, the first step, the virus has to enter the cell, so that will happen in the lysogenic cycle and the Lytic cycle both. And here, this step also, this will happen in the lysogenic cycle, Lytic cycle both. Second step. Third step is the difference point between the lytic and the lysogenic cycle. So what happens in the lysogenic cycle? From here, the lytic and the lysogenic cycle separate. So let's show the lysogenic cycle separately over here parallelly, and in the lysogenic cycle also the cell membrane would be there, it will not be vanished. Okay, and where is our cell's genetic material? Sorry, it has changed the color. Yes, here's our cell's genetic material, that will also be there, but the genetic material of the virus will not destroy this time the genetic material of the cell, but what will it do? It will do even a weirder thing like that. It will just join with the genetic material of the cell, and all this will happen, and the cell will not be able to know. Why? Because this genetic material of the virus will contain some genes inside called the repressor genes, and these type of genes will just help the virus genetic material to spy and to not let the cell know. So what happens, these genes are there, so it will just quietly attach itself to the cell's genetic material. Now in the next step, the cell will replicate by mitosis. It doesn't know that there is a viral genetic material which is also present, but it will copy the whole genetic material and then replicate by mitosis. Alright, so it will form 2 cells, and 2 to 4, 4 to 8, 8 to 16, and this process will continue in ever increasing rate. So the genetic material of the virus will also continue to replicate in an ever increasing rate. So let me copy the whole thing for simplicity. Where is my copy tool? Take it. I love the copy paste business. Take it, copy it, and paste it. Good. Too big for it, right? Anyways, hope you get the general idea. Okay, and there will be another cell also, and this process will continue, replication process, it will continue until the cell has, means many numbers cell are generated, 128, 122, so 264 I guess, 256 I get, yeah, 256, and then it will continue like this in an ever increasing rate, and the viral genetic material will also keep on replicating itself in an ever increasing rate. But suddenly what will happen? The cell will come to know that the viral genetic material is there. Why? Because some type of, every time the perfection doesn't happen and the repressor gene will fail to hide itself, and then the cells will come to know that the viral genetic material is there, but the action that the cell will take due for after coming to know that the viral genetic material is there is not appropriate. Why I am saying that, you will see in a moment. What will happen is that the cell will try to repair itself. Naturally it will repair itself, because whenever you come to know that I am not okay, then it will try, it is a natural response to repair oneself, so it will try to repair itself. How? It will separate the virus genetic material from the cell genetic material. Let's see. It will separate the virus genetic material here, sorry, the cell's genetic material here, and the virus's genetic material. Where is it? Red color, yes, there's a virus genetic material. And now again the same process of the Lytic cycle will continue. The virus's genetic material will attack the cell's genetic material, but now here the attack is in all the cells which has the virus's genetic material. While replicating itself, all the cells will continue to have the virus's genetic material. In all the cells, it will simultaneously attack the cell's genetic, all the cells, the virus will simultaneously attack the cell's genetic material, and in all the cells suddenly you will feel some disorder happening in the body, like that. So this was the difference between the lytic and the lysogenic cycle. So let me connect it with an arrow. So after that what happens is that the same process as in the lytic cycle over there, this one, okay, this one happens after that, and then this happens, that the virus cell membrane burst out and the viruses leak and attack the other cells. So that was the lytic and the lysogenic cycle. Hope you found the video useful. Alright, and see you next time, and thanks for watching. Bye.